Hello, everyone, and welcome to this session entitled CNS Disease and the Microbiome. We have three giants in the field of gut-brain research giving talks today, Jane Foster, Paul Wilmes, and Emeran Mayer, and two up-and-coming giants giving talks, Kerri Kinghorn and Elisa Menozzi. So just a couple of housekeeping notes before we begin. I'm your chair, Sarkis. I'm a professor of microbiology at the California Institute of Technology. And our session will run with three longer talks: the basic research talk, the clinical translational research talk, and the teaching talk. Those are all 15 minutes. The teaching talk is 20 minutes. Each one will be followed by a 5-minute Q&A. And interspersed between these three talks are two data blitzes of 7 minutes each with a 3-minute Q&A. For your questions, please put your questions in the chat box. I will read those questions to the speakers, and then they'll be able to answer them, but that is your mechanism for asking questions for each of the speakers after their videos play. And so again, the Q&A will be after each of the talks. There is no panel at the end. The delegates are free to discuss among themselves using the chat box. But again, sorry, the questions for the speakers have to be put in the questions box in the Q&A box. So any questions for the speaker goes in the questions box. The chat box is for discussion among the delegates. And so with that introduction, I'm very pleased to introduce our first speaker, Dr. Jane Foster, who's a professor of psychiatry at the University of Texas Southwestern Medical Center, and she's giving the basic science research talk. And the title of Jane's talk is Microbiome Crosstalk Influences Brain Development, Lessons Learned from T-cell Deficient Mice.
Good afternoon. I'd like to thank the organizers for inviting me to speak today. I'm going to tell you about some of the work that we've been doing that looks at microbiome crosstalk during brain development in mouse models. So microbes cover all the surfaces of our body. They include bacteria, viruses, fungi, protozoan parasites, across different body sites as visualized in this schematic on the right-hand upper side. There is different in composition and diversity. For example, the bacteria composition in your gastrointestinal tract is very different than the composition in your mouth, which is shown in orange for the oral. And in the end, we have 100 trillion gut bacteria. We have 1 trillion bacteria on our skin, and there's about 10 million bacteria in your mouth. And overall, this is about 1.6 times the number of eukaryotic cells in your body, which speaks to the fact that, if we have all these bacteria, perhaps they're important to our health. And when we think about the biochemical potential or the genetics of the microbiome, there actually is a remarkably expansion of microbial genes here shown as greater than 2 million in comparison to human genes, which we know from the Human Genome Project is closer to 23 or 24,000 genes. Microbiota are essential to pathogen defense. The good bacteria fight off the pathogens. They're important to nutrient uptake and metabolism. They ferment a lot of fiber in our diet and produce a whole host of important gut metabolites. They help maintain the integrity of the gut barrier. They're essential to the development and the function of the immune system. And their microbiota-brain communication is important to healthy brain development. And the focus of the work that we've been doing in my lab for the past 15 years really has focused on the essential nature of this interaction between microbes and your adaptive immune system. Several lines of evidence demonstrate that microbiota influence brain development. In collaboration with Jacob Elgood and Jason Lurch from Sick Kids in Toronto, we used high-resolution ex vivo MRI to examine the impact of microbiota status on brain volume. The figure in the middle of this slide shows the absolute volume differences between germ-free and specific pathogen-free male and female C57 black 6 mice. The total brain volume of GF mice is much smaller than SPF mice, so a better representation is shown on the right through the brain as relative brain volumes. In both of these figures, the orange or red areas are larger in germ-free mice and the blue areas are smaller in germ-free mice. As shown in the violin plots on the right, the total brain volume for male and female germ-free mice is smaller than SPF mice. Also, we see reduced relative volume for fiber tracts and increased cortical volume in both male and female mice. To determine whether these effects are reversible, we compared germ-free mice to germ-free mice that were conventionalized at 5 weeks of age and to GF mice that were colonized with a community of 8 bacteria referred to as altered Schadler flora prior to conception. The graphs on the right demonstrate that conventionalization of germ-free mice at puberty did not normalize brain volume changes, while mice colonized with ASF flora prior to conception had normal brain volume. These data and work by others demonstrate that microbrain interactions during early postnatal life are important to brain development. Exposure to microbes and colonization occurs primarily at birth and continues through development. As noted earlier, microbiome crosstalk is an essential contributor to healthy brain development. Human brain development occurs in a sequential manner, and the development of neurons, glia, and brain circuitry occurs in parallel with the development and maturation of the microbiome. In mice, the brain also develops sequentially over postnatal development as shown here. This parallels the sequential pattern of human brain development, and we can use mouse models to understand how the microbiota and the immune system contribute to brain development. Importantly, the microbiome and the immune system is developing in the same postnatal window, and we can look at outcomes in the postnatal period to understand how the system interacts to influence brain development. And seen here on the very bottom, you notice that T-cell autogeny in mice is occurring in that same postnatal window as brain wiring and microbiome maturation is occurring. Our lab has been using T-cell deficient mice for several years to examine the importance of T-cells to brain development and behavior. These mice lack all functional T-cells due to the knockout of the beta and delta version of the T-cell receptor. At the behavior level, we have shown that these mice have a developmental delay in writing reflex, increased ultrasonic vocalization in pups in response to maternal separation, reduced self-grooming. Over the lifespan, they have reduced exploratory and anxiety behavior, increased locomotor activity in the adult, losses of normal sex differences in activity, and also sex and genotype differences in brain structure, stress and immune response, as well as peripheral and central immune signaling. In the next few slides, I'll share some of the recent data looking at microbiota in T-cell deficient mice. Here at postnatal day 24, analysis using 16S ribosomal RNA sequencing, we show that the absence of T cells will lead to reduced diversity, shown as alpha diversity, in the microbiome, with both male and female T cell deficient mice shown in the red and orange compared to the blue, black 6 mice. The absence of T cells also was accompanied by compositional differences visualized on the right. This graph shows beta diversity, which considers both the composition and the abundance of bacterial taxa, and you can see that the T-cell deficient mice microbiome are clustered on the left-hand side of the graph, and the B6 mice on the right-hand side. In a recent publication, we examined the temporal trajectory of these changes, and shown here in the two aspects of alpha diversity, which increase over the lifespan. If you look at the blue plots first, that they increase between postnatal day 17 and postnatal day 24, leveling out at postnatal day 28 and remaining high through adulthood. In contrast, T-cell deficient mice have an accelerated development, and they get to a higher level of diversity by postnatal day 17, which then doesn't peak any further, and leading to a reduced overall diversity at later stages in development. On the right-hand side, we see the beta diversity for these different age categories, and again, you see the difference between the B6 and the T-cell deficient mice, but notably, P7, B6 mice have a completely different composition of the microbiome. And then there's a transition at weaning, which occurs at postnatal day 21, to a more adult-like microbiome composition, and this shift is completely absent in T-cell deficient mice. Here we show some of the differential abundance, and so there's a host of differences across both family and genus, and this bubble plot shows several specific ASVs that are increased in T-cell deficient mice on the right-hand side of the figure, and several that are more abundant in black cysts or decreased in T-cell deficient mice on the left side of the figure. We used metagenomic sequencing to confirm some of these differences we saw using 16S ribosomal RNA sequencing, and this shows those data in similar plots where we see reduced alpha diversity, differences in beta diversity, and in fact, compositional differences at the level of genus in the T-cell deficient mice. This graph just demonstrates that in those taxa that we could reliably measure both using 16S, which is shown in green, those are in 16S only, and the metagenomic sequencing shown in blue. In those ones that were shared between those two tools, we had fairly good concordance with respect to fold change between T-cell deficient and black cyst mice. Those are shown in green. In this study, we also examined both the gut and the brain metabolites to get a functional readout of the impact of lack of T cells during development. This work was done in collaboration with John Swan at University of Southampton. So we used NMR spectroscopy, and these spectrograms show the metabolic profiles of intestinal, both colon and cecal samples shown here from black cyst and T-cell deficient mice. In the representative spectrograms for cecum and colon, metabolites that are increased in TCR mice have peaks above the 0 line, and metabolites that are reduced in TCR mice have peaks below the 0 line. Notably, the gut microbiome metabolite 5-aminopentanoate, shown labeled here, also called 5-aminovalerate, was increased in cecal, fecal, and colonic tissue in TCR mice, whereas the short-chain fatty acid butyrate was reduced both in the cecal samples, also in the fecal samples, and in the related colonic tissue. To examine the association between gut microbial features and their metabolites and test for their ability to discriminate between B6 and TCR mice, relative abundance and all metabolic data obtained from the cecal samples were integrated using a supervised classification algorithm called Diablo, and the Circo plot on the upper left shows the significant association, and the plots on the right show the loading plots for both bacteria and metabolites. The graph on the bottom shows the trajectory of key taxa and key metabolites, notably for both microbes and metabolites. A subset of are increased in TCR mice, and a separate subset are decreased or absent in TCR mice. Our analysis extended to the brain, and we looked at metabolites in several brain regions, and this spectrogram shows differential metabolites at postnatal day 24 in cortical regions, again demonstrating that microbe cell interactions, immune cell interactions at the mucosal barrier influence the development of the brain metabolome. Several significant changes were observed in cortex, hippocampus, and hypothalamus, including key metabolites such as GABA and acetate, as well as other metabolites. This slide shows some of the highlights some of the key differences we observed, including the increase of Akkermansia in T-cell deficient mice, the reduction of butyrate producing bacteria and reducing alpha diversity, and alterations both within the gut and the brain metabolome. So overall, we've demonstrated in these recent studies that microimmune brain signaling is essential to healthy brain development. And we know that host factors at the gastrointestinal barrier influence the microbiotic composition and function. Included in those are T cells influence the development of the gut metabolome, both the microbe and the host metabolome, as well as the brain metabolome. And it's important that these findings are translated to the clinic, and there's some examples of some papers that we've published, one looking at microbe-immune relationships in adolescent linked to early life adversity that came out a few years ago, a recent paper looking at the immune profile associated with suicidality in adolescents, and then a recent paper that's in revision has actually identified co-occurrence networks of bacterial taxa that are associated with clinical phenotype in depressed individuals, building off this early mouse work to demonstrate that these relationships are important in clinical populations. And this is important as one of the biggest challenges in psychiatry is understanding how do we stratify individuals into more homogeneous clinical phenotypes. And by doing this, we can glean the benefits of using microbe-immune biomarkers in psychiatry. And this is because our microbiome is our own. It's influenced both by your host genetics and your life history. And biomarkers related to the microbiome might subgroup clinical populations as visualized in the previous slide, screen individuals for potential microbe-host or drug interactions, match individuals to the best treatment options, as well as identify individuals at risk for early intervention, provide novel targets for drug development, as well as expand microbiome-targeted therapies such as diet, prebiotic, probiotic, and other interventional strategies for psychiatric illness. The next 10 years, we expect to advance precision medicine approaches, and this schematic just sort of suggests that by leveraging these microbiome tools in association with tools that measure host phenotypes such as brain imaging, metabolomics, as well as metagenomics, we might be able to generate algorithms that will help predict who might respond to available therapies and how we might develop novel therapies in psychiatry. So I'd like to thank much of the work that I presented today was done in my lab at McMaster University, which has been there for the last 19 years. And more recently, I have relocated to UT Southwestern Medical Center and doing some of this biomarker with the team of scientists there. So thank you, and I'll see you live for questions.
Thank you, Jane, for that excellent talk. While we wait for questions to come in to the chat, maybe I'll just ask you, just zooming out from your work, what are the mechanisms by which T cells can communicate with the brain? So it's unlikely during homeostasis that there's a high burden of T cells in the parenchyma. What are the ways that we know of where T cells can actually send their signals to neurons?
So I think that, some of the exciting advances in the neuroimmune field in the past decade sort of parallel the work that's been happening in the microbiome, particularly some of the neurodevelopmental work coming out of Joni Kipnis's group, looking at how some of these T cells are in the meninges early in life and influencing the brain parenchyma through release of cytokines. There's also a bit more evidence accumulating of small numbers of T cells in the brain in healthy situations that are acting through the choroid plexus. And so that, I think, is a really interesting space that, some of these single cell technologies are starting to advance, the resolution to be able not just to measure them, to actually see what their functional readouts are. So in animal models, I think that's moving a little bit forward as well. Mm-hmm. But yeah, I think that there are methods. I think that the balance between T regulatory cells and the Th17 cells seems to be controlled a little bit by some of these microbial signals, potentially the short chain fatty acids, and on that T cell development. So I think that window of early life is a real opportunity that T cells could be gating some of that early wiring in the brain. And I think that might be the biggest impact. And then you're right, later in life in the homeostasis, I think what the data we show in mice that's important is without the T cell, the microbes actually take a different path. So again, that whole concept of disrupting the immune microbe balance early in life, we already know it contributes to allergy. We know it contributes to other it contributes to obesity. So perhaps there are some, vulnerabilities in brain development that we could get a little bit better resolution on.
Yeah, it was related, somewhat related. It was the striking finding that the T cell receptor animals don't have this prenatal or pre-weaning, sorry, microbiome, right? That they seem to go into this adult stage, which I think is pretty fascinating. We have a question
Yeah.
in the chat on slides, referring to slides 18, 19. I may have missed it, but is there another way to alter the microbiome than deleting T cells?
Well, I think that there's a host of ways we can alter the microbiome, right? I mean, I think that maternal immune activation alters the microbiome. The work, Sarkis, your group did early on to demonstrate that disruptions both in utero and in early life can change things. Antibiotics early in life can change the microbiome. I think there's it's not just the T cell deficient mouse. The T cell deficient mouse is interesting to me because it has a low level of anxiety or anxiety-like behavior. And there's a whole host of ways we can alter anxiety-like behavior, including other genes in the periphery or genes in the central nervous system. So I think when this body-brain imbalance occurs, there's both a top-down message that might be coming from the brain to the microbiome and the bottom up. So I think what we've shown in our data is it's bidirectional. But I think you're right, Sarkis, that transition between P17 and P21, I don't think it's just diet related. There's something else developmentally. And these mice have major delays in writing reflex. They can't flip over till like postnatal day 8 or 9. So they're a little bit messed up. Some of that's has to be happening in utero.
Yeah.
Right. It's the lack of T cells in the moms.
Yeah, no, it's
contributing to that.
It was a pretty striking finding, and its implications on the metabolome. I think the lack of the T cell receptor or T cells on the metabolome, I think, is interesting.
Well, in that paper is the metabolite data as well, so somebody could dig into that. It's in the supplemental. We did the metabolomics on those mice as well in the same tissue and in the brain.
And I wonder how much of it is immune versus just gut health, right? If it just changes GI health and changes the metabolites flowing across the intestinal barrier.
Yeah, in previous work, we haven't seen any alterations in barrier integrity or in, histological look of the gut. They seem pretty healthy.
Terrific.
So I think that it's an interesting model from that perspective.
Wonderful. Thank you again, Jane, for the excellent work. And we'll move on to our next speaker in the Data Blitz. This is Kerri Kinghorn, who's a group leader at the University College London. And the title of her talk is Intestinal Autophagic Defects, Gut Microbiome Dysbiosis, and Immune Activation Mediate Gut-Brain Axis Dysfunction in a GBA1 knockout model of Parkinson's disease.
Hello, my name is Kerri Kinghorn and I'm based at the UCL Institute of Healthy Aging. And today I'm going to be talking about what we've been learning about the role of gut and immune dysfunction in communication between the gut and the brain in neurodegeneration, and specifically that linked to mutations in the GBA1 gene. Firstly, I have no conflicts of interest to declare. So my group used the fruit fly Drosophila melanogaster to model mutations in Parkinson's disease genes. Most cases of Parkinson's disease are sporadic, but approximately 15% are familial. And most of the genes that have been linked to Parkinson's disease appear to map to endolysosomal pathways. So these are the pathways within the cell that are responsible for trafficking proteins around and ultimately leading to their degradation and recycling. And at the centre of these pathways is the lysosome, and normal functioning of this organelle is required for normal degradative processes to occur, such as autophagy and phagocytosis. And here I'm going to be focusing on one lysosomal gene called GBA1 because it's been linked to neurodegeneration and specifically to Parkinson's disease. The GBA1 gene encodes the lysosomal enzyme glucocerebrosidase, or GCase for short, and this is responsible for the hydrolysis of glucosyl ceramide to ceramide and glucose. And biallelic mutations in the GBA1 gene cause the commonest lysosomal storage disorder known as Gaucher disease, This is characterized by the accumulation of glucosylceramide within macrophages, which then become deposited in various tissues such as the liver, spleen and bone marrow to cause organ dysfunction. And rarely the brain can be involved in Gaucher's disease in types 2 and 3 neuronopathic forms. As well as causing Gaucher's disease, many multi-center genetic studies on Parkinson's disease populations have shown that heterozygous GBA1 mutations are the commonest genetic risk factor for Parkinson's disease, conferring an approximately 20-fold increased risk. And we are particularly interested in communication between the gut and brain in Parkinson's disease. And in keeping with the fact that gastrointestinal dysfunction is very common in Parkinson's disease patients, increased bowel inflammation as well as gut wall leakiness and an alteration in the gut microbiome have been identified in Parkinson's disease patients compared to healthy controls. Moreover, the communication between the gut and brain is likely to be complex, both via direct neuronal communication and also by systemic mediators such as hormones, cytokines and inflammatory cells. And in order to study the role of gut-brain communication in neurodegeneration linked to GBA1 mutations, we used a fly model completely lacking the GBA1 gene. So we first looked at the brains of GBA knockout flies, and we noticed that there was clear evidence of neurodegeneration as demonstrated by loss of normal photoreceptor morphology in the retina compared to control flies. There was also glial activation in the brains of GBA knockout flies with an increase in a glial phagocytic marker called Draper, both on immunostaining and on Western blot analysis. Just as in the brain, there was also an increase in systemic innate immune activation with an increase in 2 NF-kappa beta signaling innate immune pathways, the TOL and IMD pathways. So we then looked at the gut wall of GBA knockout flies in detail, and what we saw was that there was clear impairment in the process of autophagy in this tissue. So this is the cellular process which is responsible for the degradation and removal of misfolded proteins and unwanted cellular constituents. And what we saw is that there was an increase in a protein called Ref2P, which is usually removed by autophagy in the gut wall on immunostaining and also in the gut tissue on Western blot analysis. And in keeping with the gastrointestinal dysfunction which is often seen in Parkinson's disease patients, we also saw a constipation-like phenotype with a decrease in the number of fecal deposits produced in the first few hours after eating a meal. There was also increased gut wall leakiness with an increased proportion of the guts of GBA knockout flies being permeable to an ingested blue dye. Interestingly, we also saw an increase in NF-kappa beta innate immune signalling in the gut tissue of GBA knockout flies, as well as an alteration in the bacterial composition of the intestinal microbiome of GBA knockout flies compared to controls, as shown here on this heat map. We therefore wondered what would happen if we completely eradicated the gut microbiome by raising flies under germ-free conditions on a cocktail of antibiotics. And remarkably, this reversed immune activation in the brain, in the gut, and in the whole body of the GBA knockout flies. This led to improvement in locomotor function, and also the GBA knockout flies raised under germ-free conditions live longer than untreated flies. We also reversed the autophagy defects in the gut wall of GBA knockout flies by feeding them with rapamycin, which stimulates autophagy. Rapamycin treatment reversed the autophagy defects in the gut wall, rescued brain neurodegeneration, and also returned both systemic and gut immune activation towards control levels. These benefits were also accompanied by an improvement in locomotor ability as well as increased lifespan. And what was particularly interesting was when we treated GBA knockout flies under germ-free conditions and also with rapamycin. And in this case, we saw no additive effects on the lifespan of the flies. Putting all of this together, we have shown that defects in autophagy in the gut wall cause immune activation in response to exposure to the gut microbes, and that either reversing the autophagy block with rapamycin or completely eradicating the microbiome can dampen down both the gut immune system activation and also the systemic inflammatory response, thus reducing neuroinflammation as well. And thus the gut microbiome and intestinal autophagy defects are potential therapeutic targets for the treatment of Parkinson's and Gaucher diseases, and the use of rapamycin should now be further investigated with a view to clinical trials. Thank you very much for listening. And I would like to end by thanking members of my group and also my funders.
Terrific. It's really interesting. And I have a couple of questions that maybe we can just briefly go through. It's a full body knockout of the GBA in the fly, or is it intestinal specific?
No, it's
and have you done intestinal specific then?
Yeah, so it's a full knockout, just as it would be in patients. So it's knocked down throughout. We have tried to do tissue specific manipulations, but it wasn't really possible because the RNAi lines that were available didn't work. So we haven't performed specific manipulations where we knock down GBA, but we have performed specific manipulations where we try to genetically rescue by knocking down the immune system in the gut.
Okay. And then again, just really briefly, so the sequence of events are the gut autophagic dysfunction, changing the microbiome, changing of systemic immunity, that leads to neuroinflammation, neurodegeneration, and then are there motor phenotypes in the fly, and is it because of these systemic and brain or systemic and nervous system changes in immunity?
Yeah. So yes, the GBA knockout flies do show an age-dependent locomotor dysfunction, and if we dampen down the immune system through removing the gut microbiome, that improves the motor function. And if we reverse the autophagy defects in the gut, but also systemically throughout the fly, that also improves locomotor performance as well.
Perfect. And does neurodegeneration coincide with the motor deficits?
So flies don't have alpha-synuclein, but they do have sort of the equivalent of P62 and other proteins that aggregate and accumulate when there's autophagy defects. Yes, we do. We do see essentially neurodegeneration, though. It's not specifically of dopaminergic neurons, but we see whole brain neurodegeneration that correlates with the locomotor decline.
Terrific. Maybe even a better model than some mice, right, where there's a disconnect between neurodegeneration and motor phenotypes. Thank you so much, Kerri, for the talk. Our next speaker, who will be giving the clinical translational research talk, is Paul Wilmes. Paul is a professor at the Luxembourg Center for Systems Biomedicine and also at the University of Luxembourg. And the title of Paul's talk is Systems Ecology of the Microbiome Brain Axis.
Hello. I would like to first of all take this opportunity to thank the organizers for the ability to present today on systems ecology of the microbiome brain axis. I do not have anything to declare. Now, the gut-brain axis, and in particular the role that the microbiome may play in this, has of course drawn quite a lot of attention over the past few years. And so there are compelling links linking the gut microbiome either through propagation via the vagus nerve or via the circulatory system to central nervous system diseases. And what is important to stress in this context is that this may indeed be related to different molecules that might be produced in the gut that might then trigger pathogenic processes, again, via the vagus nerve or indeed via the circulatory system. And what we're dealing with here is, of course, a very complex situation whereby there are many different molecules that might be involved in triggering disease processes. And we have basically started to summarize all of the different molecules that might be involved in this in the context of a map or knowledge graph, which we term the expobiome knowledge base. Important in this context is the fact that the collective of these molecules, which we term indeed the expobiome, may be triggering different pathogenic processes along different pathways, such as, for example, immune pathways in the context of a range of different diseases, including neurodegenerative diseases. And so what the aim of our work is really to basically do microbial systems ecology. So what we believe this is focused around is basically the systematic integration of metagenomic, metatranscriptomic, metaproteomic, and metabolomic information, thereby covering all of the different omic levels and also covering all of the different molecular classes that may indeed play a role in triggering disease processes. And the aim of this is, if we do this in a proper systematic manner over both space and time, we should get to the point of being able to reconstruct constituent networks of individual microbial populations, and also, of course, importantly, understand how the emergent properties give rise to molecules that may, of course, be very diverse, ranging from nucleic acids all the way to small molecules that themselves then, of course, may trigger disease processes in susceptible human cell populations, such as, for example, of course, in the context of neurodegenerative diseases, neuronal cells. And so basically, we've had an interest over years now in the context of the microbiome and its link potentially to Parkinson's disease. And from some of our early work, we found, in fact, that specific microbial taxa are enriched in the context of Parkinson's disease, not necessarily across all individuals, but certainly popping up in certain ones. And importantly, these organisms are typically linked to specific functional guilds, meaning that they basically are responsible for certain functions in the gut. And one key organism which we found to be enriched in Parkinson's disease is the genus Akkermansia, of which the representative taxon is Akkermansia muciniphylla. And as the name already suggests, there is a link to, indeed, mucus in the gut. And so from this initial finding, many different groups, in fact, have been able to replicate this. And they have found also Akkermansia to be enriched in Parkinson's disease patients. And this is, in fact, linked to a higher propensity for mucin and host glycan degradation, whereas at the same time, you see a depletion in starch and other complex polysaccharide degradation. And so this was interesting to us because we had, in fact, observed higher levels of Akkermansia, in particular muciniphylla, in the context of a completely unrelated work where we were interested in contrasting fiber-rich to fiber-deprived diets and see what effects this switch in diet may have, in fact, on basically the microbial community. And here we used a synthetic microbial community, and then what effects that might have on host physiology. And what we found is that in the fiber-free or fiber-deprived diet, there was an enrichment then in Akkermansia muciniphylla, which in turn led to basically foraging of the mucus layer, its thinning, and making mice under a fiber-deprived or fiber-free diet much more susceptible to infection by an enteric pathogen. So what we here are interested in then is this thinning of the mucus layer combined with exposure to, for example, some form of a toxic molecule may in fact then trigger Parkinson's disease in the gut, whereby you then get propagation mostly via the enteric nervous system and the vagus nerve. So basically, what might be different toxic molecules that may be playing a role here? The most compelling one is, in fact, a bacterial protein, which has been linked to, in fact, alpha-synuclein aggregation. So this is basically Kerri. Kerri is a protein which is expressed by Escherichia coli and clearly has amyloidogenic properties in that it can act as a seed for amyloid protein aggregation and fibrillation. And based on Sarkis' lab's work in a THT assay, where alpha-synuclein was co-incubated with Kerri, an increase in alpha-synuclein aggregation velocity was indeed observed. So this means that Kerri indeed can act as an amyloidogenic protein in the context of alpha-synuclein aggregation and fibrillation. So in a study that we then performed, combining both the dietary aspects as well as exposure to Kerri, we basically used a Parkinson's disease mouse model carrying a wild-type alpha-synuclein transgene under the regulation of the Th1 promoter, which leads to predominant neuronal expression. And we exposed this also then, of course, to a fiber-deprived diet, as well as Kerri expressing E. coli, or indeed the combination of both, including, of course, also the respective controls. The treatment period was a total of 9 weeks, where at baseline we also recorded sensor motor behavior. And then we also, of course, collected fecal samples over time, and also performed overall health monitoring of the animals. So in the subsequent week, we switched basically the diet in order to prime the microbiome also for the following bacterial gavage with E. coli expressing indeed Kerri. So the combination of both fiber deprivation and Kerri exposure, this continued for a total of 8 weeks, during which feces were also collected and basic motor functions and health were monitored. So in the final week, the sensory motor test was repeated to detect potential exasperation, and tissues were collected, as well as, of course, as I mentioned already, fecal samples for 16S rRNA gene amplicon sequencing. And so the tissues that were collected underwent then various pathohistological analyses. So the 16S rRNA gene amplicon sequencing of the murine fecal samples that were collected then over time revealed that fiber deprivation caused a reduction in microbial diversity overall. And we also observed a shift in various genera which have been linked to barrier and mucus integrity, including in particular Akkermansia. What is also important to stress, in contrast to the previous work, is that this was not a synthetic community, but these were specific pathogen-free animals, so meaning that the overall microbial diversity was much higher than in the previous study which we had performed. So it's clear that we then have an enrichment indeed in Akkermansia and other mucus foraging bacteria in the context of fiber deprivation. And so a question then over what are the link changes physiologically in the gut. So when we then measured mucus thickness, we found that the outer mucus layer, which of course is a habitat for several commensal colonic taxa, was really very much thinned, as we had observed earlier, that in the context of a fiber-deprived diet. Furthermore, we found that phosphorylated alpha-synuclein, which is of course a marker to identify potential toxic moieties of alpha-synuclein, this was in fact enriched in enteric ganglia, and that of course links this then to the transgenic animals being exposed to Kerri, which again is expressed by E. coli. So when we next looked into the nigrostriatal pathway using immunofluorescent workflows, we observed that the combination of both fiber deprivation and also Kerri exposure led to noticeable changes in alpha-synuclein and also overall neuronal pathology, as well as also motor skills, more specifically coordination, as highlighted here on the right-hand side. So basically, to summarize our findings here, what we see is that the combination of fiber deprivation and Kerri exposure in the presence of human alpha-synuclein led to a number of recapitulations in terms of Parkinson's disease-like phenotypes. And based on these results, we propose a potential disease mechanism which involves both diet, so diet-induced dysbiosis in relation to, in particular, fiber deprivation, that indeed may reduce, for example, the thickness of the mucus, thereby making animals much more susceptible to exposure to toxic in toxins and exons such as the amyloidogenic protein Kerri. And so that, of course, first and foremost in the enteric nervous system, but then we might have subsequent propagation via the vagus nerve to trigger alpha-synuclein pathology and neurodegeneration in linked and susceptible brain regions. So this is something that is, of course, very much of interest, linking diet, lifestyle, then to potential pathogenic mechanisms linked to the microbiome. But there may be, of course, many other factors at play. It may not be as simple as diet and then exposure to a particular toxin. So we developed recently in the lab a tool called PathoFact, which aims at basically predicting virulence factors and antimicrobial resistance genes in metagenomic data. And I'm not going to go into detail here. I'd just like to highlight that we indeed applied it also to data sets from Parkinson's disease patients. And interestingly, we found in Parkinson's disease patients enrichments in vasins. Vasins are, of course, molecules that are very important for disruption, for example, of the epithelial barrier. And importantly, also vasins are linked to Enterobacteriaceae, such as Escherichia coli. Furthermore, we applied this PathoFact pipeline to another study that we performed where we were interested in antibiotic exposure. And so what is really striking from this study is that organisms that do very well after being exposed to a broad spectrum antibiotic cocktail are indeed Akkermansia and Enterobacteriaceae. So what this data suggests is that both organisms which we first implicated in Parkinson's disease pathogenesis based on our mechanistic work may in fact get a selective advantage through antibiotic exposure. So to summarize this a little bit here, basically many different mechanisms may be at play, including, of course, a thinning of the mucus layer that might be diet-driven, and then exposure to different molecules that may indeed have a direct effect on, for example, alpha-synuclein aggregation such as Kerri, or that may in fact also be further involved in the disruption of a barrier in the gut, such as, for example, these invasins that I just highlighted. Okay, so basically to conclude, so the expobiome may act as a trigger of many different disease processes, including alpha-synuclein aggregation. We have quite a lot of evidence pointing towards a microbiome-driven pathogenic process triggering Parkinson's disease. Overall, I'd like to stress that basically form or community structure follows function, and that therefore we should really focus on function in relation to a mechanism. And then, of course, subsequently, we really need to look more, I think, into lifestyle and environmental factors and how those, through the microbiome, may in fact be causal in the context of neurodegenerative diseases, including, of course, in Parkinson's disease, but also potentially in Alzheimer's. So with that, of course, many different people to thank. And I would like to thank you very much for your attention and just highlight that we have some postdoc and PhD positions available. Thank you very much.
And thank you, Paul, for your excellent talk. We have a question in the chat, and please send more questions if you have them. But perhaps I'll start. It's a very attractive model, at least to me, in terms of this relationship between Akkermansia and E. coli. We do see them co-occurring, and the mouse model suggests that there is, that each has its own role. Did you determine in the animal model that E. coli-driven effects on pathology and symptoms is dependent on the mouse being colonized with Akkermansia? Does that precede, or is that required for E. coli's pathogenicity?
Yeah, thanks, Sarkis. This is, of course, a very important question. And we haven't specifically looked at this, whereby we wouldn't have yet done colonization experiments involving just Akkermansia, for example, and E. coli, to look at whether the combination of both would indeed suffice, I guess, in triggering the hallmarks that we observe. So this is indeed an important follow-up experiment. I should stress that also in relation to the antibiotic exposure, I mean, all of this work also was done in SPF mice. And the reason for this was that we wanted to retain as much of the microbial diversity as would be needed. I mean, I think to be more representative on the microbiome side, but I agree that these singular or double colonizations would absolutely be relevant for follow-up work now. Yeah, thanks.
Yeah, especially with a perplexing organism like Akkermansia, which in some contexts has, appears to associate with beneficial effects, and in many other contexts a negative effect. We'll go to the question in the chat. So Paul, one of the members of the audience asks, for microbes associated with PD, did you find the overlap between the identified species in PD and the ones also occurring in irritable bowel syndrome and or chronic constipation, as these conditions have been reported to be comorbidities in PD?
Yeah, I mean, basically, this is also a highly relevant question, of course. You do see, of course, commonalities in some of the organisms that are enriched. I mean, also, I should also highlight that it's not as simple as just finding, of course, Akkermansia to be enriched. I just highlighted that because that was the basis for our hypothesis that we then aim to test in the mouse model, linking it to diet. But I mean, there are definitely many different taxa that have been seen to be enriched in Parkinson's disease that also pop up in IBD. But what I would say is that there is, at least to my understanding at the moment, there is not really a strong common signature. I mean, you basically have usual suspects that do pop up, I mean, including Enterobacteriaceae from time to time, but it's not like I would say that there is a common microbiome signature between Parkinson's disease and IBD.
The question, Chad, spurs me to ask another question that we've been thinking about, and in some ways have addressed in the mouse models, but still remains outstanding, is to what degree is the microbiome configuration in PD driven by constipation versus the disease phenotype versus environmental, let's say, dietary changes? Because again, it's a chicken and the egg scenario when you're doing cross-sectional sampling of individuals, early or well into their disease course.
Yeah, no, this is I think constipation is, of course, also in relation to maybe the microbiome analysis that one does might be also an important confounder of those analyses, because we of course do know that there is, for example, a link between Akkermansia, also some of the Bacteroides species that we see popping up, to longer gut residence times. So the other one organism or group that we're very interested in is, in fact, the methanogens, and also those, so archaea in that case, and also those are linked to constipation, a longer residence time. So I think this is a very pertinent question. But also there, I think there is also a compelling link to fiber, for example, because of course what is known is that a diet rich in fiber would increase gut residence time. So maybe also the constipation is somewhat again linked to the diet that these patients, or before they in fact become patients, that they usually follow. So I think there are many different dimensions to unravel there. Linking constipation then to the taxa and the disease processes that those may trigger, and how they in turn relate to lifestyle factors. That's what I wanted to stress at the very end there.
Yeah. Yeah, we showed in that 2020 paper that you highlighted that Kerri will induce at least fewer fecal pellets. I can't generalize the GI issues in the mouse beyond that, but it's much more complex in people, right? Because again, we don't know the sequence of events. There's just so many complexities of whether or not the microbiome is reshaped because of GI issues, or whether the microbiome itself is contributing to the GI issues, or likely some combination of the two. And I think that's important in thinking about quality of life for Parkinson's patients, right? I mean, if there's an opportunity to improve GI health, I think that's going to tremendously improve the day-to-day living of individuals with PD, at least the majority of them.
No, I completely agree. I think the question, of course, that we are always asking in this context is what exacerbates, I guess, the disease, and what is an actual trigger. And I think this is, I mean, we know that the microbiome is, of course, has great potential for both. But I think this is what the work that we have over the next years ahead of us is to really drill down to the molecules. And the molecules themselves can be quite disparate. I mean, you have curly such like a protein, but you also have small molecules that can also be amyloidogenic. So, I mean, it's we should not kid ourselves. This is, of course, very complex, and there are many still routes to explore here.
Paul, since I have you as a captive audience, I will ask you a question that I've been wondering about, because you're looking at these things. To what degree, it's a general question, to what degree do microbes use proteins to signal to the host versus small molecules? I mean, proteins like Kerri are very, very stable proteins. They're at the epithelium. So I can see them being mechanisms of communication. But broadly speaking, and this is again a general question, but broadly speaking, do you think the microbiome uses proteins regularly to communicate with the host because the gut is such a hostile environment for proteins? Do you think it weighs more to small molecules, lipids, and non-protein structures?
I mean, I think proteins certainly play a role, but I think where the moieties, the amino acid moieties that are much more relevant in this context are, of course, peptides and small proteins. I think small proteins, this is, relatively recent work still from Amy Batt's lab, where they found a huge diversity of small proteins. And I think those molecules are much more likely in relation to, again, amino acid moieties to play a role in intercellular communication in the microbiome. But then the other thing that I think is also is relatively unexplored is basically biosynthetic gene clusters that are oftentimes also horizontally transferred. And here there is also tremendous diversity, so leading to many, many different small molecules that are then involved. And I think your question is, of course, highly relevant, because another chemical class that is a little bit ignored so far is nucleic acids. Mm-hmm. They also may play an important role in intercellular communication in the context of the microbiome. And I think here, we are at the moment trying to get at least to resolve the extracellular molecules. We've just optimized our integrated multi-omics methodology to specifically look at, nucleic acids, peptides, proteins, and small molecules to get at this question: what is the relative importance of those molecules? I mean, in relation to possible intercellular and then, of course, interspecies, inter-kingdom communication.
Paul, thank you again for an excellent talk. We will move on to our next speaker, Elisa Menozzi, who will give us a data blitz talk. Elisa is an MD from the University of Modena in Italy, and she is currently a PhD student at the University College London. And the title of Elisa's talk is Metagenomics Analysis of Gut Microbiome in Parkinson's Disease Patients with and Without GBA1 Gene Variants.
Good morning and good afternoon, everyone. It is a real pleasure for me to present you today the preliminary results of this collaborative work between UCL and INRA in Metagenopolis, which is supported by the Aligning Science across Parkinson's. This work is investigating the metagenomic composition of the gut microbiome in patients with Parkinson's disease, with and without variants in the GBA1 gene. Patients with Parkinson's disease who carry variants in the GBA1 gene represent 10 to 15% of the Parkinson's disease population worldwide, and they tend to show a more severe clinical phenotype as compared to sporadic Parkinson's disease patients, in particular those individuals who carry severe variants in the GBA1 gene. However, we don't know which factors or markers can drive this phenotypical difference. Alterations in the composition of the gut microbiome have been detected in patients with Parkinson's disease, but again, whether these alterations can contribute to clinical symptoms in Parkinson's disease is still unclear. With this work, we therefore wanted to address two key questions: whether the gut microbiome composition differs in individuals with Parkinson's disease with and without a GBA1 variant, and whether a specific composition of the gut microbiome might reflect differences in the clinical phenotype. To answer these questions, we had enrolled from our UCL cohort patients with Parkinson's disease carrying or not a variant in GBA1 and healthy controls, mainly spouses or partners of patients with Parkinson's disease. From each participant, we have collected extensive clinical information and stool samples. On stool samples, we have performed an in-ray metagenopolis, shotgun metagenomic sequencing, which allows us to accurately detect and quantify microbial species and estimate species richness up to the strain level. These are the demographic features of the cohort included in the current analysis. 136 individuals have been enrolled, of which 44 healthy controls, 69 sporadic Parkinson's disease patients, and 23 GBA-PD patients. The three groups are matched in terms of age, sex, and years of education, which was an important requirement for our study. And nearly half of the individuals with Parkinson's disease participated into the study with a partner or a spouse. Considering the two PD groups, patients are homogeneous in terms of age of onset, disease duration, disease severity, as measured by the Yonen-Yar stage, and levodopa equivalent dose. At the time of this analysis, only 11 sporadic PD and 2 GBA PD patients were drug naive. From a clinical perspective, the only clinical feature that was different between sporadic PD patient and GBA PD patient was sense of smell. As reported in the graph on the left-hand side of the slide, the mean score of the APSIP test, which measures olfactory function, was significantly worse in the GBA PD population as compared to the sporadic PD population. All the other measurements did not differ between the two groups, in particular the MDS-UPDRS total score, which is a measure of overall disease severity, and the Wechsler constipation scoring system, which is a measurement of constipation. The first microbiome feature that we analyzed was microbial species richness. As shown in the graph on the left-hand side of the slide, microbial species richness was not significantly different between healthy control and Parkinson's disease groups. The second analysis we performed was a classification of samples into enterotypes. Enterotypes are clusters of microbial communities, and based on the relative abundance of microbial species, we assign samples to three main enterotypes: the enterotype Bacteroides, here represented in red, the enterotype Cribotella, represented in green, and the enterotype Firmicutes christensenellales, represented in blue. The overall enterotype distribution was different across the groups, with the predominance of the red enterotype Bacteroides in the healthy control group and the blue enterotype Firmicutes christensenellales in the Parkinson's disease groups. Based on these findings, we then asked ourselves which were the factors that could have affected such difference in enterotype profile. What we found was that individuals with predominance of the blue enterotype showed higher levels of microbial species richness compared to the red and the green one. Also, individuals with predominant blue enterotype suffer from more severe constipation as compared to the other two. This is also summarized in this principal component analysis plot based on enterotype distribution, where we showed also that microbial species richness and constipation and autonomic dysfunction severity contribute to the abundance of the blue enterotype. Species analysis is currently ongoing, and these are only preliminary observations based on the limited sample size available at the time of analysis. By comparing healthy controls represented in yellow and Parkinson's disease patients with and without GBA1 variants considered together, here represented in purple, we found 13 contrasted species, all enriched in the healthy control population. Of these, some species like Roseburia intestinalis and species belonging to the Ruminococcus genus have been previously shown to be reduced in Parkinson's disease. The species Bacteroides vulgatus has also been shown to have protective effect against LPS-induced intestinal inflammation, so potentially it might also have a role in Parkinson's disease. To summarize, our preliminary data show that patients with Parkinson's disease and variants in GBA1 have more severe impairment in sense of smell as compared to sporadic Parkinson's disease. The distribution of enterotype differs between healthy controls and Parkinson's disease patients, but GBA1 status does not seem to have an effect. Finally, the enterotype Firmicutes christensenellales, which is predominant in the Parkinson's disease population, is associated with more severe constipation and autonomic dysfunction and higher microbial richness. Thank you for your attention.
And thank you for a wonderful talk. We have time for maybe one or two questions. First question is, why do patients with GBA1 mutations in the Parkinson's cohort show a similar phenotype compared with sporadic PD?
Thank you. That's a very good question, an important one. One important premise is that in this cohort that I have presented here, so 23 patients with Parkinson's disease and GBA1 variants, nearly all of them had either risk variants or mild variants in GBA. So only 3 of them were carrying a severe variant. So clearly we are sort of missing the more extreme phenotype. And also linking to Kerri's work, actually, so we know that, for example, the effect on nearly all had variants or mild variants in GBA. So only 3 of them. Thank you. Okay. So we do not really know about the effect of risk variants, for example, on GK's activity. We know that is not really impaired. So this clearly could have an effect not only in neurological function, but also at the gastrointestinal level.
Mm-hmm. I think your work is fascinating because if we're able to assign specific microbes to particular mutations or families of mutations in Parkinson's, I think that opens up many mechanistic studies to look at what I think is central in PD, and that's gene-environment interactions. So thank you very much for your work, Elisa. And we'll move on to our final speaker, who is giving our teaching talk, Emeran Mayer, who is a professor of gastroenterology and neuroscience and is the distinguished professor of medicine at the University of California, Los Angeles, and he's also the executive director of the Oppenheimer Center for Neurobiology of Stress and Resilience. Emeran's teaching talk, and he gives wonderful overviews, is entitled The Challenge of Translating Brain-Gut Microbiome Science into Humans.
Thank you for inviting me to the Brain Conference and to give a short presentation on the challenge of translating brain-gut microbiome science into human diseases. None of the entities listed, I have no conflict of interest for the current presentation. So microbiome science obviously has revolutionized our understanding what factors influence overall our human health and what role they may play in disease. The realization that our mere 20,000 human genes are totally outmatched by the up to 20 million microbial genes. Opens up a whole set of questions of how a understanding of this large number of microbial genes and their products, what role this plays in our health and also in our disease. So let me start out with a couple of slides emphasizing that what's at the bottom really of the challenge of translating this exciting microbiome science into human diseases with implications for diagnostics and therapeutics. So this has to do with the multifactorial architecture of human brain disorders, which in my opinion requires a systems biological approach. And that is the fact that this has not generally happened with animal models is probably at the bottom of this problem of translation. Our microbes, the gut microbiome, shown at the bottom here, is linked with our brain via multiple bidirectional communication channels, either directly from the microbes to the brain or indirectly via the immune system and endocrine system. We also are in close connection with the exposome, the environment, and this system in humans is constantly perturbed by psychosocial factors, including stress, but also by dietary perturbations in form of unhealthy diets. So in understanding how individual microbes might play a role in this complex system is obviously very difficult, maybe even impossible, because it involves so many factors that participate in this brain modulation. A kind of simplistic model to link the gut microbiome to the brain is shown here, where we have on the right lower side the gut connectome, a system of interconnected immune cells, neuroendocrine cells, and epithelial cells and nerve cells, which interact with the gut microbes and their products, the microbe-derived molecules. But in addition, the gut itself communicates with the brain via multiple channels, both top-down via the vagus nerve, but also bottom-up via these multiple communication channels. And then we have a direct pathway as well from the microbes to the brain. So I'm showing this because it emphasizes that our traditional models from microbiome science do not quite take this complexity into account. We're still so driven by identifying particular microbes or particular mediators, molecules, as being causative in terms of very complex human brain disorders. And hopefully throughout this talk, I'll emphasize that I don't think that is really possible. So several approaches have been taken to link the animal models to, and what we have learned in microbiome science, to causality in the human setting. And I've summarized sort of a few of these attempts. So starting at the top, the majority of available studies today are these epidemiological and cohort studies where we compare the microbiome in a disease state with the microbiome of a healthy control state. There are many of these studies, but none of them really have proven causality for the microbes. Another one is that we compare phenotypes, so human disease phenotypes with preclinical disease models in the mouse. These comparisons of phenotypes, again, do not prove causality, but depending on the validity of the mouse model, provide intriguing hypotheses that there may be a link between the two. So there's a growing number of studies recently where fecal material is transferred from a human disease state to into humanized mouse models. And there's a growing number of these studies, and in general, they seem to be confirming the fact that there's something in the fecal material of human disease states that can induce a pathological phenotype in the mouse. And on the other hand, many attempts trying to link or trying to implicate a fecal microbial transplant from a healthy donor into an individual with a particular disease have failed. The only exception is the C. difficile colitis situation, but all the other attempts have really not been successful. I mean, there can be multiple reasons for that, but it certainly has not given us the another proof for causality. And then another possibility would be that you take patients with different diseases, you have a therapeutic intervention aimed at the gut microbiome, and if this resulted in a healthy phenotype, then this would be obviously an ultimate proof for a causal role. This type of intervention, unfortunately, up to date, has been largely limited to dietary intervention studies. So despite these limitations, there's been an explosion of publications, speculations, mirrored in the lay press, that have implicated alterations in the gut microbiota, often summarized in the concept of gut health, with multiple not only brain diseases such as Parkinson's, depression, Alzheimer's, autism spectrum, but also with a variety of other metabolic conditions and even malignancies, cardiovascular disease. And it's been shown that there is an interaction. There seem to be correlations between these various diseases. And there's also fairly good evidence that an inappropriate activation, engagement of the immune system, but potentially originating from the gut, may play a shared role in the pathophysiology and etiology of these diseases. So let me go through sort of a couple of models of, or a couple of ways of illustrating what we currently know about this relationship between the human diseases and the microbiome and alter brain function. So as I just showed you on the previous slide, the association studies, epidemiological cohort studies, have identified a large number, shown on the right in the right box. A large number of diseases which have now been referred to as brain gut microbiome disorders, ranging from schizophrenia to anorexia nervosa. None of these have really been proven, even though for some of them we have pretty intriguing evidence. A good example, two recent big studies which showed the power and limitation of these large-scale association studies, both published in Nature Communications. The first one came to the conclusion that bacterial genera associated with depressive symptoms belong to multiple families, prominently including the families of these genera listed here. In the second one, found an association of 13 microbial taxa with depressive symptoms. And it's interesting what the authors concluded, that these bacteria are known to be involved in the synthesis of glutamate, butyrate, serotonin, and GABA, which are key neurotransmitters for depression. And they conclude that this study suggests that the microbiome composition may play a key role in depression. That's obviously a speculation at this point, despite the high quality and the size of these two studies. But it shows the limitation really of this type of studies in terms of identifying causality. So then there's a variety of mouse models of human brain disorders showing a dysbiosis. So this is now implicating in the mouse that an alteration of the microbiome produces symptoms that we consider as disease models for human disease. How strong this validation is, that these actually reflect the same thing as the human disease, we don't know, but it shows you that a variety of these mouse models have been shown to be correlated with altered gut microbiota. And then evidence that in these mouse models the microbiota play a role has been shown from interventions to the gut microbiota manipulation in these mouse models, so with probiotics, with antibiotics, fecal transplants, SPF, which have shown a change in the in the strength of the mouse phenotype. So these studies have really shown significant evidence, indirect evidence for causality, even though the key link or the key proof we've not been able to establish. So let me go through a list of advantages and limitations of mouse models to understand and manage human brain disorders. So the advantages of the mouse models are they allow interventions that are not possible in humans to study the causal role of gut microbiota in health and disease. They have the comprehensive knowledge of mouse genetics and availability of numerous genetically modified mouse models more than any other models. Mouse models are inbred, so providing a homogeneous genetic background, cleaner system to dissect signals from gut bacteria host interactions and improve reproducibility of experiments. And you'll see that this also comes up as one of the main disadvantages. And then sources of variation, such as diets, housing conditions, are generally controlled for in experiments, limiting unwanted influences of noises from the surrounding environment to gut microbiota. And you'll see that these two latter points are really also the main disadvantage or challenge for translation to human disease. So the limitations, despite important similarities, mice differ from humans in brain anatomy, both in terms of the brain, like, for example, the size of the prefrontal cortex and the importance in disorders like anxiety and depression, also the enteric nervous system, genetics and physiology, and therefore cannot fully recapitulate human systems. The crosstalk between gut microbiota and the host is host specific, so observations in mouse models might not be applicable in humans. And here comes one of the major factors. So the genetic homogeneity implies that the inbred mouse strains cannot capture the inherent genetic variations in the human population. The variability introduced by, the inter-individual variability introduced by genetics in humans is significant and is something that has been eliminated in the mouse models where these very clear-cut results have been obtained about relationships of microbes with the brain. Another very important factor of anybody who is involved in human studies realizes that multiple factors, such as genetic background, birth mode of delivery, mode of feeding, menstrual status, diet, medical history, multiple psychosocial factors, all contribute to shaping a real-life gut microbiota in humans. An absence of these factors in mouse studies, where we have successfully been able to eliminate those, implies that the gut microbiota in these models cannot reflect a real-life gut microbiota. In my opinion, this may be one of the most important reasons for the failure to date. So the take-home points, this very brief overview of the challenges of translation. There's no question that the communication between the gut microbiome and the gut-based immune system is an ancient evolutionary conserved system that modulates the function of every organ, including brain structure and function. Reductionistic preclinical research has revealed remarkable bidirectional interactions within the brain-gut microbiome system, resulting in structural, functional brain changes and behavioral alterations. This is the exciting reality of the microbiome science to date. But in contrast, cross-sectional epidemiologic studies have demonstrated associations between gut microbial composition, function, and behaviors without convincing evidence for causality between the gut microbiome and the brain. And there's multiple theoretical and biological barriers to explain this discrepancy between animal findings and clinical relevance. Certainly one of those reasons that I mentioned is the factors in humans that play a role influencing the brain to result over the lifetime to produce distinct brain phenotypes and behavioral phenotypes is much larger than it is in the mouse model, and we will just not be able to overcome this discrepancy. So anybody who is interested more in about some of these concepts and the human translation into brain-gut microbiome disorders, I refer to my website and to the 2 books I've written on this topic. Thank you very much for your attention.
Emeran, thank you for the overview of the field and obviously this burning question of to what degree do animal models represent human biology and what are the translational outcomes of microbiome research, which I'd like to ask you about in 1 second. Maybe I'll start with the question on just theoretically, what have mouse models told us about the microbiome in terms of its potential relationship to human disease? My personal feeling is that since there's just such strong evidence in mice that the microbiome is likely causal in so many different conditions, that is very unlikely to not reproduce in humans. That I don't see any reason why the mouse would be such an outlier in terms of its dependence on the microbiome versus other organisms. That being said, the mechanisms by which the microbiome may communicate in a mouse and a human, or the effects it may have between the 2 species, are likely very different, right? Do you agree with that premise, right? That there's something that we've identified in mice, which again suggests that there's a likely role in humans. But more importantly, what is the translational path forward? I mean, if we want to really leverage all the work that's been done, ultimately we have to ramp up human studies. And what do those look like?
Yeah, I mean, I totally agree on what you just said. I mean, it's one of the most amazing parts of recent science, I think. This whole, the linking the microbes in multiple ways now to brain and behavior. I have to say, I speak from painful experience in the first part of my career when I focused on visceral pain models and spent a lot of time and found dramatic effects of therapeutic candidates in the mouse model, which did not translate into the clinical trials. So I sort of came to this with the goal to really prove it in the human situation. I quite honestly, other than the things that I said, I don't have a good explanation. I think it will ultimately happen. One sort of simplistic way of looking at it is if you have so many influences competing, with how they shape the brain, the brain-gut microbiome system, maybe it's that the microbe component to the variability is 5%, whereas so it's very hard in a heterogeneous population to demonstrate that. I guess similar with genetics. I mean, we're running into this in multiple areas of science, and Yeah, it's a frustrating situation, but I'm not sure. So just as you said, I'm totally convinced there's something very fundamental and substantial in these relationships. But the challenge to translate this into clinically meaningful effects is very difficult, I think. I mean, I don't know what the other.
Yeah, no, it is, but certainly of high reward, right? And if you think about, I mean, just broadly, genes versus environment, and if in this case, environment is the microbiome, I would argue that manipulating the microbiome, at least in 2023, is much more straightforward than manipulating the genome. So it presents opportunities. It presents at least the opportunity to begin testing some of these hypotheses in people. And the FMT studies, as you highlighted, have been disappointing. But I also think that it's a lot to ask of a fecal transplant to ameliorate symptoms in a very complex disease. I think we need to be a little bit more strategic and also have interventions where the effect size is larger. Emeran,
just a quick comment. Just talking about diet. So in human studies, so we know, everybody knows that diet questionnaires are totally unreliable, almost no correlation with actual food intake. A very important component in the microbiome. So this is just one example which hopefully with the development of foodomics techniques, we will be able to overcome this and really know what a patient eats and then correct the microbiome findings. And then there's several others of those purely technical limitations, that you do clinical trials. So many have been published, for example, without sex differences. So there's a lot of examples that, so I think it will be a matter of time, in my opinion, before that proof can be delivered and specific therapeutic interventions can be based on that. That's sort of my personal view.
Thank you for that, Emeran. And with that, with those profound words, we will close the session. Thank you, everyone, for joining the CNS and microbiome session. And again, I want to thank the speakers for absolutely wonderful talks and to the delegates for your attendance. Thank you.